Welcome to this lecture on cell biology. Today, we are going to explore a fascinating and vital process called facilitated transport. Imagine your cells as bustling cities surrounded by high walls. How do they get the food and supplies they need without breaking the wall or spending all their energy? Understanding this mechanism is fundamental to understanding how life itself is sustained at the microscopic level. Let us dive in and uncover how cells solve this critical problem. To understand our topic, let us start with a simple analogy. Imagine you are trying to get into an exclusive concert, but the venue is surrounded by a massive, impenetrable brick wall. You cannot simply walk through it. However, the venue has special revolving doors and VIP escorts designed to let certain people through safely. In a cell, the plasma membrane acts just like that impenetrable brick wall for many essential nutrients. Facilitated transport is the biological equivalent of those revolving doors and VIP escorts. It ensures the cell gets exactly what it needs without expending any of its own energy to pull them inside. Now that we have an intuitive picture, let us define this formally. Facilitated transport, which is also known as facilitated diffusion, is the process by which specific molecules move across a biological membrane. Crucially, they move down their concentration gradient, meaning they go from an area of high concentration to an area of low concentration. Because they are just flowing down this natural gradient, the process is entirely passive. The cell does not need to spend any of its precious energy, or ATP, to make this happen. However, these molecules cannot do it alone. They require the help of specialized transmembrane proteins to cross. This brings us to an important question. Why do these molecules need help in the first place? Well, in simple diffusion, small, non-polar molecules can just slip directly through the lipid bilayer of the cell membrane. But life requires larger, polar, or charged molecules, such as glucose and amino acids. The core of the cell membrane is highly hydrophobic, meaning it strongly repels water and water-soluble molecules. If a polar glucose molecule tries to cross, it is immediately pushed back by this water-fearing core. Facilitated transport is the elegant solution to this problem, providing a shielded pathway so these vital nutrients can bypass the hydrophobic barrier entirely. So, how exactly does the cell provide this shielded pathway? It relies on two main types of specialized transport proteins embedded in the membrane. The first type are channel proteins. You can think of these as open, water-filled pores or tunnels that allow specific ions to flow straight through. The second type are carrier proteins. These are a bit more complex. Instead of just being an open tunnel, a carrier protein actually binds to the specific molecule on one side of the membrane. Once bound, the protein changes its physical shape, shuttling the molecule across and releasing it on the other side. Let us look at this visually to make it perfectly clear. On the screen, you see the cell membrane, represented by the phospholipid bilayer. On the left is a channel protein. Notice how it forms a simple, straight pore. The small blue ions are flowing directly through it, moving down their concentration gradient from top to bottom. Now look at the right side. This is a carrier protein handling a larger glucose molecule, shown as a green hexagon. See how the protein is open to the outside to catch the glucose? Once the glucose binds, the protein undergoes a conformational change. It physically shifts its shape to open toward the inside of the cell, safely releasing the nutrient. Both methods bypass the hydrophobic core completely. Now that we understand the mechanism, We must ask, why does this matter so much? The truth is, this process is absolutely critical for your survival. Consider your brain. Your brain cells rely almost entirely on glucose for energy. But as we learned, glucose is far too large and polar to cross the membrane alone. Without specific carrier proteins called GLUT transporters, your brain cells would rapidly starve, leading to unconsciousness and death. Furthermore, the rapid movement of sodium and potassium ions through facilitated transport channels is exactly what allows your nerve cells to fire and send electrical signals. Without this system, our cells would be completely isolated and unable to function. Because facilitated transport relies on physical proteins, it behaves very differently from simple diffusion. In simple diffusion, if you keep adding more molecules outside the cell, the rate of transport will just keep increasing indefinitely. But facilitated transport is limited by the actual number of doors available. As the concentration of the molecule increases, the transport proteins eventually become fully occupied. They are working as fast as they can. This leads to a strict maximum transport rate, a phenomenon known as saturation. No matter how much more substrate you add, the speed cannot increase further because every single transporter is busy. This saturation behavior is perfectly described by a famous mathematical model called the Michaelis-Menten equation. Let us break down what each part of this formula means. The variable v represents the actual rate of transport happening right now. Vmax stands for the maximum possible rate of transport, which occurs when every single protein is busy. The term S, in brackets, represents the concentration of the substrate, which is the molecule waiting outside to be transported. Finally, Km is the Michaelis constant. It represents the exact substrate concentration needed to reach half of the maximum speed. If the substrate concentration increases massively, the rate V approaches Vmax. Conversely, if the cell loses transporter proteins, Vmax drops, slowing everything down. Let us see this formula in action with a concrete example. We are looking at a human red blood cell using the GLUT1 carrier protein. We are given that the maximum transport rate, Vmax, is 100 picomoles per minute. The Michaelis constant, Km, is 1.5 millimolar. The current glucose concentration outside the cell is 3.0 millimolar. We need to find the current transport rate. We start by substituting our known values into the Michaelis-Menten equation. In the numerator, we multiply 100 by 3.0 to get 300. In the denominator, we add 1.5 and 3.0 to get 4.5. Dividing 300 by 4.5 gives us our final answer of 66.67 picomoles per minute. Physically, this means the transporters are working at about 2/3 of their absolute maximum capacity. Now, let us mathematically demonstrate the concept of saturation using the exact same cell. Imagine the person just ate a massive, sugary meal. The blood glucose concentration spikes dramatically to 15.0 millimolar, a 5-fold increase. The physical transporter properties, Vmax and Km, remain exactly the same. We plug our new concentration of 15.0 into the equation. Multiplying the numerator gives us 1500. Adding the denominator gives us 16.5. Notice how the denominator is now heavily dominated by the substrate concentration. Dividing 1500 by 16.5 yields a rate of 90.9 picomoles per minute. Even though the glucose concentration increased by a massive factor of 5, the speed only increased from 66 to 90. This physically proves that the carrier proteins are nearly saturated. They are working as fast as possible and cannot exceed their limit of 100. We have covered a lot of ground today, so let us summarize the key takeaways. First, facilitated transport is a vital, energy-free mechanism that allows essential molecules to cross the cell membrane. Second, it relies on specialized channel and carrier proteins to shield these molecules from the membrane's hydrophobic core. Third, because this process depends on a finite number of physical proteins, it is subject to saturation, meaning there is a strict maximum speed at which molecules can enter the cell. Finally, we saw how the Michaelis-Menten equation perfectly models this behavior. Understanding these concepts is fundamental to grasping how cells maintain balance and survive. Great job following along, and keep exploring the wonders of cell biology.